Okay, hi everyone. Thank you so much for coming. This is our second annual mock study section. A few things to get started. You all hopefully received the grants that are being reviewed today, or at least a portion of the grants. These were graciously given to us by current and former postdocs. So please be respectful with their material and don't just disseminate this widely. With the exception of the K99, which is freely available on, Katie, can we turn that off, please? Which is freely available from NIH, the National Institute of Aging. So by all means, you can access that and share that in any way you might want to. To get us started, big thank yous to all of our faculty volunteers, some of which are here now, others will be coming later today. For Katie Payne for helping us get all of this set up. There's coffee and pastries in the back. Feel free to help yourself throughout the morning. Oh, it's over here. Okay, our schedule for today. We're starting with an F32, which is an NIH grant that is available to postdocs, although international postdocs largely are not able to apply for that one. Then at 11, we will go to a VA grant, a Career Development Award 1. We have lunch at 12, and then at 1 p.m. we will review a K99, which is a grant that is accessible to all international postdocs and postdocs from here. And then we'll finish off with a Q&A session. So if you have some questions, please feel free to write them down so you can ask them at that time. We are running this as like an educational event. So at certain points, our faculty will stop and explain things or go into more depth than what you would normally see in an actual study section. So just know that this might look a little bit different because of that. But to get us started with the F32, we have our chair, Dr. Julie Christensen, reviewer number 1, Dr. Anna Gorsica, reviewer number 2, Dr. Sufi Thomas, reviewer number 3, Dr. Liskin Swint Cruz, and then serving as panel members, Dr. Dan Tilden, Dr. Lee Chavez, Dr. Matt Morris, and Dr. Michael Van Son. So thank you all for being here. Thank you, faculty. Julie, I'll turn it over to you.
Okay. I don't know how, do we need to hold this probably close by? This is fun. Just like in a regular study section, we have to use our microphone voices. Okay, so I'm going to give a little background about how we would have gotten to this point. It's not just magic that your study section members appear in a room. So most of the fellowship applications will be reviewed by a standing panel. So there are a number of fellowship standing panels that exist within the Center for Scientific Review, or CSR, which is the designated review section of NIH. Occasionally you will have fellowship applications that are reviewed within an institute itself, but generally it's going to be within CSR. And so there will be a scientific review officer for each of these panels. I'm going to kind of do a hybrid version of being an SRO and the chair for our mock study section. So the SRO works for the Center for Scientific Review or for an institute, and is responsible for recruiting reviewers, assigning applications, and is the designated like NIH official at the study section meeting itself. And so all of the applications come in, they're assigned to, they go to CSR, they're assigned to an institute, they're assigned to a review panel, and then the SRO will receive those applications. They're going to be standing members of the study section. So these are faculty who've been recruited and they have specific terms. They will be at each meeting that happens for however long their term is. And then there'll be ad hoc members. So these are members that, depending on the type of like the science that's being reviewed, if there are, you know, like too many applications for a specific institution and you have a lot of conflicts within your panel, you might need to get additional members coming in. So the SRO is responsible for recruiting those additional members to be on the panel. Once they have their panel set, they will assign the applications. And so that occurs through, I don't know how many of you have experience with ERA Commons, which is such a delightful website. Love two-factor authentication. And so these will be assigned, the reviewers will have to basically affirm that they are not legally, what is it? You're not a lobbyist. You're not a federal lobbyist, which is kind of funny because I don't think any of us are. But you have to say that you're going to maintain confidentiality, and then you'll get your applications. You also go through to make sure that you don't have any conflicts with anyone. So if you've got, so a conflict would be if I've published with somebody in the last 3 years, if I have a close personal relationship with them, if it's somebody at your institution, you're considered a conflict and you cannot review that application. You cannot be in the room when the application is discussed. You don't see the application in Commons. You don't see the critiques. You don't see the scores. You see nothing. And so you'll receive your applications, you're given a due date, you read the application, you write a summary, you also have the summary statements. That's essentially what we write. And then we input scores. What you won't see in the summary statement is the overall impact score, which is what I will ask the reviewers for when we start our discussion. That's kind of it's not an average, it's more of a conglomerate. So some scores are going to be worth more than others. In a fellowship application, the individual, the candidate themselves is going to be the most heavily scored because this is an application to fund an individual. It's not to fund a research plan or a research program. You're not giving money for supplies or anything like that. You're giving money to support this trainee. And so that is the most important. And then the training, the sponsor, and usually the science is lower at the bottom. Versus an RO1 where it's science, investigator significance, kind of thing like that. And so, yeah. And so you put in your scores, you show up at study section. It would be more of a big long table where we're all facing each other, but you don't want to look at our backs. I don't know, maybe you do. And so we'll start there. Okay. And lights go down. And now we're in the scene. Okay, so here we are. We're at study section. The SRO is going to say, welcome. I'm Dr. So-and-so, the SRO of this meeting. We are here to discuss however many applications. Today we're here to discuss one F32 application. Everything that happens within this room is confidential. Please do not share anything that happened. That is for perpetuity. You can't talk to somebody 10 years from now and be like, hey, I actually reviewed your F32. It was great. Can't say that. And so the SRO will then say, everybody introduce yourself. We'll go around the room. Okay. I'm the chairperson. I'm Dr. Julie Christensen. I'm a professor of cell biology and physiology at KU Med in Kansas City. I'm a basic scientist. I study the impact of early life and lifelong stress exposure on the development of urologic pain disorders and comorbid metabolic and mood disorders as well.
I'm Lee Chavis. I am an assistant professor, PhD general bench scientist. I'm in internal medicine, division of geriatrics. I study epigenetic changes in frailty and aging transitions.
I'm Dan Tilden. I am a physician scientist. I am an endocrinologist, both pediatric and adult endocrinologist, and I study the transition of care and ways that we can support young adults as they move from pediatric to adult care, particularly rural young adults with type 1 diabetes.
I'm Anna Gorsica. I'm an assistant research professor in the Department of Internal Medicine at the University of Kansas Medical Center. My research is in physical activity epidemiology and the dissemination and implementation of lifestyle interventions to underserved populations.
I'm Liskin Sweet-Cruz. I'm professor and chair of the Department of Biochemistry and Molecular Biology, and I study protein evolution in order to build better predictors for personalized medicine.
I'm Sufi Thomas, Associate Professor of Otolaryngology at the University of Kansas Medical Center. My focus is primarily on the role of the tumor microenvironment in head and neck cancer progression and response to therapy.
I am Michael Van Son. I'm an assistant professor at the University of Kansas Medical Center in the Department of Cancer Biology. My research focuses
peripheral energy state is transmitted to the brain and controls energy homeostasis.
Okay, now that we know who each other are, as chair, I would be frantically writing down if there's any names that I'm unfamiliar how to pronounce, I would be writing that on. I would have a roster and I would be writing that down. Just a little peek behind the curtain. And so now I will say as chair, like, you know, I would give instructions on how I would like to lead the meeting. So I'll say, we're going to be discussing however many applications, approximately 15 minutes for each application. This means reviewer one should give their general overview, just a brief synopsis of the grant itself. You know, be sure to talk about the individual, the candidate, the training of the candidate that's been proposed. And then, you know, focus on those really score driving items that you noticed in your review. Okay, so to get started in the discussion itself, the SRO would say, okay, now we're going to discuss application number, whatever. Okay, so this is an F32. The PI is Harrison Sarawalt from the University of Kansas Medical Center. The title of the application is Exercise-Induced Regulation of CYP7A1 and Bile Acid Metabolism. The candidate sponsor is John Tifo. Are there any conflicts? Are there any conflicts in the room? We are all conflicted because it's from our institution. But if there were any conflicts, if it was an in-person meeting, they would be asked to leave. If it was a Zoom meeting, they would be kicked out. Like you're put in like a holding room and then they bring you back in once it's over. Okay, so then I'll say our reviewers are Dr. Anna Gorsica, Sufi Thomas, and Liskin Swint Cruz. Could you please state your preliminary overall impact scores?
4.
3.
4.
Okay. Dr. Gorsica, could you please give your overview of the application?
Sure. The PI has a lot of publications for the stage that he is at as a first-year postdoc. He's looking to investigate the effect of exercise on bile acid synthesis. The study, the sponsors and collaborators seem to be well-versed to help the postdoc achieve these goals. The proposed studies are relevant for human health and have a very translational significance. There were many strengths for the applicant, specifically being highly motivated, strong publication record, and the excellent mentoring team and collaborators in the field of the study proposed. The training plan was fairly well presented. However, there were some issues mechanistically with the process of basic science. That's all I'm going to say about that. And specifically how there was a process that was denoted as one single process, but actually it's 2 separate processes, which was one of the biggest issues with the training plan that I had, as well as the evidence provided in the training plan was conducted in rats and the proposed research was to be done in mice, and those are 2 different animals.
And I'll just interrupt to say that we provided the reviewers with the actual reviews that were done on this grant. So Dr. Gorsica is pretending to be this reviewer one, even if she personally disagrees with some of the reviewer comments.
All right. Thank you. I agree with the reviewer one for the most part. I think my score was a little better because of the strength of the mentoring team and also the strength of the candidate. So this candidate has 9 publications to date, 3 as the first author. The mentoring team is extremely strong, very good track record of extramural funding, a number of very experienced investigators, including Dr. T4, Dr. Lee, and Dr. Burgess. They're all experts in bile acid metabolism. The training plan is excellent. It has a number of exciting and novel areas of interest. The experiments are very well thought through and described. However, there is a minor weakness because of which I didn't give a better score. My main concern is that AIM-1 is not mechanistic enough. It is somewhat descriptive in that, for example, in AIM-2, they did propose knocking down or reducing the levels of key genes in this particular project that would enable them to understand better the role of that gene in the system, whereas AIM-1 is not well developed enough, and they do not propose these siRNA-based experiments on this particular protein. And so that was the major weakness and concern that I had about the research training plan. But the training potential is excellent. This proposal really gives them a very good toolbox to take into the future as a scientist. In addition, the mentoring plan and the career development plan will really aid the candidate in their future endeavors.
I agree a lot with what reviewers 1 and 2 said. The candidate is an extremely strong candidate. Because they're such a strong candidate, I think that it would be good for them to have another round, because what they're lacking in their research training plan are alternative outcomes, not just alternative techniques, and they haven't considered alternative hypotheses. So one of those that I can think of is that exercise could reduce the production of excess acetyl-CoA, and that would change the net flux into both cholesterol, bile acids, and DNL. So I think they need to spend some time developing alternative outcomes and alternative hypotheses, and also provide some rationale for some of their studies that it's not provided. In their training potential, I think their integration with the co-mentors should be strengthened. They're proposing to meet just quarterly, and if those get skipped, one gets skipped, then you go a long time without meeting. So I think they should be meeting more often in order to make that an effective collaboration.
Okay, this application is open for discussion. I'm wondering if one of the reviewers, even if it's somebody on the panel, wink, wink, Dr. Morris, could give a little more insight on the application itself and what they're proposing. Dr. Conflict at the end of the table.
Well, I mean, overall, they're assessing whether you can dump or waste energy, excess energy, through the production of cholesterol and ultimately bile acid, and whether exercise actually accelerates this process or increases the production of acetyl-CoA that is then funneled towards cholesterol and ultimately bile acid synthesis. And hopefully this would be a mechanism behind how exercise can improve energy homeostasis and maybe weight loss or preventing weight gain. But some of the problems that I have with it is the way the experiments are laid out. It's like a 2x2x4. And I think it's, even if they were able to pull that off, it would be underpowered at 8 to 10 mice. So, I mean, they could break those experiments up and still get to the same questions, answer the same questions. But more globally, my biggest issue is that they just say they're just going to use C57 Black 6 mice. They don't tell us what substrain. So there's no way for them to address any potential issues with any of those substrains. Particularly in this case, if they're using Black 6Js, they already have a SNP that reduces their mitochondrial capacity or makes them more susceptible to mitochondrial disease or pathophysiology. So it's really hard for me to accept this just saying they're going to use Black 6 mice. We need to know what they're going to use so that we can actually interpret better how the data might come out.
Thank you, Dr. Morris. That was a really good recap of the application. But in, support of this candidate, it being an F32, and I get it that there are minor issues with the experimental design. And like I also mentioned, they don't use siRNA in AIM-1. However, looking at the strength of the candidate and also the strength of the team, a mentoring team, I really think this is a strong application and worth, scoring high.
Can I ask the SRO a question?
Sure.
How much should flawed science of the research plan impact the overall score of the candidate for F32?
A bit of a judgment call for the reviewers themselves. I think we can see if everyone has the summary statement, like they don't.
No, they do not.
Okay, so overall, like this application got really high. So there's 5 criterion scores that you also enter in as a reviewer, as well as an overall impact score. And it's it's the candidate, I have these. Okay.
The fellowship applicant, the sponsors, collaborators, and consultants, the research training plan, the training potential, institution, environment, commitment to training. And then the overall score.
And then the overall score. And so the criteria that you're asked to give scores on don't match exactly the components of the application that you're reviewing. So when you review an R01 or like an individual award for faculty level, you're scoring significance, innovation, approach, investigator, environment, and these are all parts of the application. And so it's really easy to suss those out. But for these, it's fellowship applicant, which is kind of, there is a candidate statement, but it's weaved throughout. The sponsors, yes, but research training plan. So that is the research plan that's submitted, but it's also if the experimental design is flawed, how well is that training going to be? Like if you're being trained to do an experimental design that's not complete or is underpowered, how good is that training? So it feeds into that. And then the training potential also falls under that. It's kind of this overall, like, how good is the candidate? How good is the sponsor? How good is the environment? What is the potential for this person to be trained? And again, if you see significant, I don't know if we have these fatal flaws, as we call them, in grant applications as reviewers. If it's one of those, then yeah, it's going to go down. But if you think that through the process, we're always told to review the grant that we're given, not the grant that we want it to be, which can be hard, especially if you're familiar with their research. So, you can take, you shouldn't, but sometimes as reviewers, you take for granted that they'll be doing the experiment and figure out that they're underpowered, or, oh, I should add this control group because these results are not interpretable without it. But again, you have to review what's in front of you.
I think the panel that I just served on, so I just served on an F panel in March. I think really it was the research training plan that was driving the scores as much as anything. And I finally put myself in the headspace of if I were on their, say, a graduate student committee, would I be advising the student to take this route or not?
Yeah. The problem is once you get to the applicants for these awards are already the cream of the crop. Like everybody got into graduate school, everyone got into a competitive postdoc. You're already dealing with the best of the best. And then to be discussed in the meeting, you're the top 50% of the best of the best. So everybody's already like, I date myself. They're all from Lake Wobegon. They're all better than average. Like nobody in this audience understands that joke, but everybody up here does. So they're already exceptional individuals. The sponsors are already super. So it does sometimes fall on that research plan that's what differentiates an excellent proposal from an okay proposal.
And I would say as a first year, brand new, not even a year into postdoc, I would send this back for training purposes to develop those alternative hypotheses.
Yeah.
All right, I'll chime in too. So we've actually submitted 4 F31s in the last year and a half, and every time they've come back, it's always the training plan is always the weakest link of all of them. You know, we get ones and twos and everything else, but training plan is always 4 or 5. But then when you resubmit it, that's, you know, the good thing is that's an easier, I think, thing to fix than some of the other ones. And a lot of the comments I think that we got towards that training plan too were communications with the specific expert that is going to give them that specific training, right? So, and I think, you know, if you read through this one too, you know, they talk about all these experts and all the expertise they have, but it's not clear that they've ever actually met with that person and discussed what they're actually going to do for them. And so we get a lot of, you know, we had a lot of those comments.
Yeah.
So I think just improving that too will help improve the training plan in pieces as well as the training.
Yeah. Okay. Well, okay.
Unless.
No.
I think the other piece that I'd say is that you want to be thinking about, the thing that I find missing in this application was what's your pathway to that next grant, right? How are you going to get the K? And these are the skills that I need. And so not only are you painting the picture of what my current research is, but this is going to lead me to be able to do this next grant that's going to work for my K. And so I think that was one of the things that could really strengthen the training plan piece of this is really laying out that roadmap for reviewers to be able to say, okay, this is going to set you up to be able to do this next step, because they're not funding this particular research plan as much as they're funding you as a researcher, as an investigator, and they want to fund your career. And so that's something that I find missing in this application was just that clear pathway to the next step.
Okay. We're going to go back into character now. We need like a light that shows when
Yeah.
breaking the fourth wall. Okay. Any other discussion on this application? Okay. What about additional review criteria? Vertebrate animals, were those okay?
Yeah, sorry.
Yes. Okay. Any?
Can I do a sidebar?
Yeah.
Change the light again? So, this reviewer also did not find any issues with vertebrate animals. However, if you look at the criteria for vertebrate animals, there are 4 sections to it. The 4th one is euthanasia. The plan for how the mice are going to be euthanized has to be in there. And neither in their A0 or A1, they had the 4th criteria. So they're missing one of the 4 criteria. But I don't know if for exercise physiology, this is not required. But the NIH website states that you need all 4 criteria addressed.
Only if it's outside of the norm. No? You need it? Okay.
Yeah. So I don't know, they didn't pick. No, it has to be
In the vertebrate animals.
animals section. No, good. I'm glad you brought that up. That's great. That's interesting. Okay.
Oh, do we? Okay.
No, but thank you for raising that point, but no, it has to.
State that on the microphone, because hers wasn't.
Yes, there was someone in the audience that mentioned that the form of euthanasia was listed in the research strategy portion. And the panel is just clarifying that it also needs to be in the vertebrate animal section.
Thank you.
The mice are from Jackson, by the way. It's in the vertebrate animals.
Look all the way at the end.
Yeah. Okay. Any biohazards listed?
I'm so sorry. Okay, no problems with the biohazards. However, this is new criteria for the NIH, and this is just being hammered into us right now, is you have to have in your facility section somewhere about how you're going to handle biohazards, biosafety level 2, or whatever the other. And none of us do it, okay? Majority of the grants that I'm reviewing do not have it, and then it's listed as unacceptable. So please, just want to point that out.
And this was new And this is a new application. Yes, okay. So if that is all the discussion, I will summarize our discussion. So this, if I could find it, this is a new application from a first-year postdoc at the University of Kansas Medical Center, working with John Tifo. The underlying premise of the grant is to address whether you can increase energy expenditure through the generation of bile acids and whether exercise can enhance this process, and for a means to improving energy homeostasis. The PI was considered to have a very strong publication record. They have 9 papers, 3 as first author. They're considered to be highly motivated. Mentoring team is excellent. John Tifo is an established extramurally funded investigator. They've recruited 2 additional co-investigators with relevant expertise and experience. The proposed studies were considered to be novel and relevant to human health. The training plan was well presented, had a number of exciting and novel areas of interest, generated a good toolbox for moving forward into an independent research position. There were concerns about the meeting schedules with the other investigators. That was considered a minor weakness. The biggest concerns were within the research plan itself. One major weakness was noticed that there was a conflation of 2 separate mechanisms that were being presented as a single mechanism. That was a big concern of the application. AIM 1 was considered to be generally descriptive, could use more mechanism within it, although AIM 2 was considered to be mechanistic. There was not a good consideration of alternative outcomes and their potential impacts on the study. The experimental design was also considered to be underpowered, and the lack of mention of which strain of black mice they were going to use within the application itself was considered a weakness. Overall, there was moderate enthusiasm, good enthusiasm for the research team itself, but concerns with the research plan dampened the enthusiasm in the room. Anything else to add? Nope. Okay, so if I could please get your final scores, viewer 1.
5.
I'm going to stay at a 3.
4.
Okay, the range is 3 to 5. Please input your scores. So at this point, we would all be in commons.
Ask if anybody's voting out of range.
I'm going to, just calm down. So at this point, we would input our scores in commons, and the SRO would say, is anyone planning on voting out of range? So if you are even 1 point, you have to state, raise your hand. And if you were not involved in the discussion, so like Matt could raise his hand and we would be like, yeah, he shared his piece. But if Lee here, he's been very quiet. If he raised his hands, we'd be like, would you care to elaborate?
Well, I found the applicant highly motivated and the research plan thoroughly put together, and I'm voting out of range.
Fair enough. Okay, vote your conscience. And then you vote. And then scores are in, and I say, okay, what about the budget? Any issues with the budget? There generally aren't because it's a set budget. Resource sharing, authentication, select agents, foreign justification. Okay. This concludes application number 1. And then the conflicts would be let back into the room and we can't talk. Even like, is that still working? Okay. Even if if there's a related application later on, say somebody else is looking at bile acids, a reviewer can't say, oh, unlike that other application that we reviewed earlier, this is looking at, you can't say that because that gives too much information for that conflict. Or you can't say, yeah, when I was this reviewer on this grant, you don't mention it at all anymore. Okay.
Can I ask a question?
Yeah.
Okay, so the 3 reviewers are the ones giving all the information, but everyone on study section is voting.
Everyone on the panel will vote.
Okay.
And as chair, I didn't mention it here. One of the biggest powers you have as a person on the panel is that vote. So if there's a range, and I hate when everyone gives the same score, it drives me crazy. So it's like 3, 3, 3. Fantastic. Great. So we're all voting 3. That's what causes score compression. So grants are scored based on the percentile. So you get this impact score, and then it's based on, I think, the previous 3 or 4 rounds, like where it falls in with the previous scores, you get a percentile, like where it ranks among all the other grants. And that is usually the pay line that the institutes use to determine who's getting funded and who's not. So if everyone gets a 3 or a 2, like then you end up with this tiny little range for scores. What you want to do is use the whole range. So 1 is the best, 9 is tears. Nobody's happy with a 9. And so what you want as a reviewer is to use that range. So if it's like, okay, I personally thought that was a really great application. I know these, I know them in the field. I don't know them personally. I think this is really important science. I think it should be funded. I would vote a 3. If I'm like, this is stupid. I don't think this was very good. I agree with Matt. This is a terrible model to use. I'm gonna give it a 5. The worst thing you can do is give it a 4, is like hit the middle, because you're not swaying anybody's opinion. You're having no impact on the vote. So you want to vote on the margins, on the edges, to have the biggest impact. Yeah, Danny. Uh-huh.
We just have someone in the audience commenting that put the information.
Not always. We need the boxes that you throw.
All right, I was just saying that when you're preparing an application, make sure that any information relevant to a particular section is present in that section. So the issue about the pentobarbital not only needs to be in the body of the application because it describes the research plan, but it also needs to be in the animal statements. The other issue about the science, just be aware that if you're, that can be reflected in the mentors. I've heard it said 1,000 times. Well, the mentor didn't read this, otherwise they would have clearly made the applicant correct it. Katie and I were having a discussion about this earlier in the day, and I've seen it both ways. The recommendation's been made by the committee and the mentor and ignored by the trainee. So it's often reflected in the mentor has not read the application carefully and so on. And the last thing for this one, nobody discussed really the letters of reference. And since I have chaired 2 panels in the past week, letters of recommendation, both panels. People provided draft letters of recommendation that they're morons who signed them just copied and pasted. Everybody got panned for that because the letter writers phoned it in. Provide letter writers bullet lists of things and make them write the sentences in their own language, or it will kill the grant. So, sorry.
No, those are excellent points. Thank you. Brings up a good point up here. So when we're in the discussion itself, we only discuss the top half of the grants that we're in. So they're ranked by score, by these scores that are put into the system beforehand. So only the top, and that's going to be like in these panels, it tends to be F31s, F31 parent awards. So just straight up F31s will be discussed together. The diversity F31s will be discussed together. The F32s will be discussed together. So like half of each of those batches will be discussed. But any reviewer, so if a reviewer feels very strongly, like you can end up with like the primary scores being 2, 4, 7, and then it ends up averaging down below. But if the 2 feels really strongly that this should be discussed and they've looked at reviewer 3's critique and they're like, they're completely off base, they missed this important part, you can rescue it. So you either tell the SRO ahead of time, be like, hey, I want to discuss this application. And then they'll discuss it.
I was just wondering, there's another new criteria come out on data management that has to be not just included in your grant application, but also as a line item in the budget. And even if you budget 0, you still have to have that item. And what that means is, whether you're going to put all your data in a secure server or in the university that has some oversight or ethical issues or what. I guess, Danny, I don't know if you have any comments on that. And I'm not sure if it is applicable for F32s, but you guys might want to just look into that.
The panel doesn't see it though, the plan.
Right.
It only goes to program.
The panel doesn't see the plan, but needs to be included in the application. However, the panel does have to check that there's the line item in the budget.
Yeah.
Okay, so what we wanted to do next was to show that this grant did not get funded on this first try. And so the grantee resubmitted the grant. And when you resubmit a grant, you get 1 page for these NIH grants called an introduction, where you get to address all of the reviewers' concerns. And so you should have received this in your packet. So you can just make note of how the grant writer went through and addressed some of these concerns. So we can see questions about power calculations are addressed here. Clarifying about the multiple pathways, which were brought up by reviewer 1, addressed here. Over here, they talked about now making the AIM-1 more mechanistic by targeting this insulin signaling pathway. And then also address this concern of the advisors in training, folks not being in like great working relationships, active working relationships. So hopefully that helps give you an idea of how you would then proceed once you receive the summary statements and reviews. And just to go into a little bit more detail, if your grant is not reviewed, so it's not discussed, you still receive the reviews from the 3 primary reviewers, and then you address those concerns in this introduction. And so in reality, this grant was not discussed on its first submission. They received the 3 reviewers' critiques. They resubmitted with this introduction and the accompanying changes to the grant and the application, and then the grant was funded on that resubmission. Is there anything else anyone in the panel wants to bring up with that?
There we go. Okay. So when you submit a revised application, that is part of the discussion of the revised application. So then I'll say, when I was asking for additional review criteria, I said vertebrate animals, biohazards, and then I asked if this was a new application. So if it was a revised application, then I would say, were they considered, how did they respond to the previous round of critiques? They are not allowed to say, well, I saw this first time around and this was my problem. You cannot say that you were the previous reviewer. Again, that if somebody is in the room that was there before, now they're in you could have new conflicts that arise, and that now somebody is like, oh, well, I didn't realize you reviewed that. So like, you're just giving away too much information. So you can't talk about, and when you get a revised application to review, all you get is the summary statement. So you only get those comments from the prior reviewers. You don't see the first submission. So you're kind of going off of what they're saying in this introduction, and you're not even required to mark the text anywhere in the grant where you changed it. I think it's nice to just put like a line in the margin so that your reviewers can see, oh, this is different. You're not required to do that, but it can be helpful, especially if you are a repeat reviewer, because that does happen. You're almost better unless it's reviewer 1, because it was a meanie.
I'm not an expert.
I tend to be the meanie, so it's fine.
But Julie,
So I had to
Yeah. No, I get it.
But you can change the color of the text to show them what you've changed.
You can.
You can.
You can.
It's
a little busy.
It looks horrible. It look, sometimes I italicize what they change.
Or they'll change the font. I like a line.
You can also say like where it's changed, like see research strategy on
Yeah.
page, like blah, 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 versus the lines in the column.
Yeah. I've seen like, we're gonna, like, changes are in Georgia font. You're like, please don't do that. Please don't change the font. It gets to be too much. Yeah.
Just don't use Georgia.
Yeah. Oh, I could go on a rant about formatting.
Oh, while Julie's talking, don't use the word utilize ever.
Unless you're actually utilizing something.
Well,
Look up the definition before you use it.
Are there any questions from the audience?
The mics are on, so you should be able to.
Yeah, okay.
Oh, okay.
I had a question. So there was one weakness regarding alternative hypothesis. So could you please elaborate on that? What was the major weakness and how to improve that?
So when I read the first version of the grant, I actually looked for, they had alternative techniques and approaches, but you also need, what are the expected outcomes? What outcomes are going to support your hypothesis? And what are alternative outcomes? And if you get the alternative outcome, is it going to support your hypothesis, or how is your hypothesis going to change?
Okay, so that should be mentioned on the main hypothesis part.
Now that's it. I like to put it at the end of each aim in the research strategy. Now we've done all this work. What do we expect to see? Is it what's going to support the hypothesis? If we see something different, what would that tell us? What direction would that move us?
Is that also? I'm responding to that. Is that also more like potential at the very end of the application when you say like issues that might arise? Potential pitfalls. Is that where you would also put that?
I often people, when they do potential pitfalls, it's technical pitfalls. And so I have started chunking it out as a separate expected and alternative outcomes, pitfalls and alternative approaches.
Which admittedly can be hard in a 6-page application to get all of that adequately explained.
And it's some of the hardest thinking, and I really struggle with doing it and put it, because I know in my gut, this is the right experiment to do. But then to make yourself think through what you might get it is tough.
And there's no set, like, how each section has to look. People will approach it in entirely different ways. They may convey the same information, but it's not necessarily in the same order or sections or anything like that. So, yeah, it's fun.
Get it in front of a lot of people.
Yeah.
To get eyes
Yeah. Are we audible?
I think so.
Okay, I have 3 quick questions. One, how do you evaluate the motivation of the candidate? Because if it is based off of publications, then someone from a big lab would have, there's possibility that they'll have multiple publications as opposed to someone who's small lab. That is one. And the second question is there any words or maybe phrases that are frowned upon that reviewers don't like? Oh, I don't like to see the word utilized, or just an example, something one. So yeah, let's get that.
It's just me.
I think the letters are really big for the motivation, and the letter should be describing this person came in and drove this project. This person came in and sat down and read the literature and devised this idea. They should also in these be addressing who developed the question. Did the advisor develop it and give it to the trainee, or did the trainee go sit down and scrounge the literature and come up with this idea? And that both the trainee and the advisor in an F grant should state that.
To Danny's point earlier, saying how, you can have an application, it's like, clearly the mentor didn't read this, there's no oversight. You can also have the other problem of, clearly the mentor wrote this, it could be too good. So it's this Goldilocks that you have to hit with almost everything. Yeah. And my other point, use dumb words. So if you want to use a fancy word and there's a dumber word that you can use that will get your point across, use the dumb word.
To that point, at least one of the 3 reviewers is not going to be an expert in the area. So jargon should be avoided at all costs. And a trigger word in every study, or 2 trigger words in every study section I've been on. Exosomes, when they mean extracellular vesicles, because they haven't defined them. And novel, which by definition means refreshingly new. 99% of the time people use the word novel, and it's not refreshing, nor is it new. So you need to make sure that you don't just throw the buzzwords in. Every cancer grant says, oh, this will lead to the cure of cancer. No, it won't. Okay, it may, but it's not likely to. So avoid those kind of platitudes.
On the dumb words idea, not only is the reviewer sitting and having to get through 6 or 8 of these, so you need to keep it as simple as possible. It's highly likely you'll have somebody who's not a native English speaker. And even if you talk to them and they seem wonderfully fluent, they never have this, it's impossible for them to have the same extent of vocabulary. So you do need to keep the simplest words and maybe run it through somebody who is doing science in a second language, which blows my mind that anybody can do that. Because there's, if you're a native English speaker and you read a lot, which many of us do, you might pick up this wonderful vocabulary and you have to go put it on the back shelf. Can't use it in science writing.
And not to be a Debbie Downer, but back to your comment about motivation and if you come in with a lot of publications versus not, like Julie said, it's the cream of the crop and then it's 50% of that. So if you don't have 9 to 10 publications, like I was, as a postdoc, I had a, different background and it was like, don't even, focus on your research question, focus on really understanding the research. And I didn't go for an F32, but now I have an RO1. So, everybody takes a different path. I know this is specifically for F32s, but the applicant, publication, your team, everything like that really has to be in the right area to be successful.
It's also area specific. I mean, if you study aging, you're not going to have 9 or 10 papers. So hopefully one of the reviewers realizes that, you're a strong candidate because you work in this area and you have 2 papers. Yeah. Or you have 10 because you're in a different area. But hopefully one of your reviewers will be able to set everybody straight if they get this person hasn't published very much.
Or if you actually have that, one of your letter writers should make a note of, in this field, it's really hard because of this, and this is really good publication rate.
Yeah, have your letter writers address like any perceived weaknesses that you think your reviewers might see. And specifically, if you're a first year postdoc, you've got 3 papers that you're waiting on your graduate mentor to read and get out. If you have a good relationship with that mentor and they're writing you a letter, which they should, have them say, wholly my fault. It is my fault that these papers are not out there. I'm working on them. He's going to have 3 first author papers come out next year. Like have them fall on that sword for you. So, and a good mentor should.
I always look for at least a review article. If somebody has not scanned the literature and at least, as an outcome of their literature review, written a review article, that's the least that you can do, in my opinion. Although some people say that you have to be an expert to write a review. I don't fully agree because, your mentor is the expert, but you can put in all that effort to gather literature and put together your thoughts. That counts.
We have one more quick question in the chat asking, what is the typical size of the panels?
It's usually 30 people. It varies depending on how many applicants the panel gets every cycle. The cycles that I'm at, sometimes we get 150 to 180 applications. So it's a big panel.
You can find the study sections on CSR's website. You can even just Google award study sections, and you should be able to get to the link. You can also use NIH Reporter and use their search function and put in like whatever application you're going to apply for, like F32, mitochondria, exercise, hit, find, and then you will find all of the funded current F32s. And you can go in each of those and find out which institute's funding it, what study section reviewed it, who their program officer is. And so that can unearth a whole wealth of information for you.
And in area comments, you get the full roster.
Awesome. If you all will please join me in thanking our faculty panel for this first review. We'll take a brief break and then we'll start at the top of the hour with our next grant.